How come I get an introduction? Yes. I think nobody told Francesca that we're not supposed to introduce.
Yeah, there's no introduction.
There's no introductions.
I've been doing it all day, okay.
Yeah, exactly. Excellent.
I didn't get the memo. Okay, David, take it away.
Perfect. Excellent. Well, this is my third talk here, so I definitely didn't need. It took that long to be able to get an introduction, you know. But in any case, but if I keep trying, yeah, exactly. Okay. Also, by the time the third talk rolls around, you start to realize the time-saving tricks that I use when making talks. For instance, recycling opening slides and all my transition slides and stuff like that. So, giving away all my secrets. But in any case, this is a topic that I am very passionate about, Earth movers distance on particular spaces, graphs in particular. And I'm going to be using it to compare microbial communities. Now, both of my previous talks have been about microbial communities. And I'm going to be figuring out how to take two of them and see how similar or different they are. This talk goes chronologically, and I struggled a whole lot with how I wanted to present this and whether we were going to cover this first or that first, and I started retooling the slides all last night, and then I forgot to save it, so we're just sticking with this order right here. But in any case, quick rehash, what is metagenomic comparison? Well, I've shown slides like this a couple times before, but you take an environmental sample, soil, earth, human body site associated, and you get a whole bunch of short sequences from it. And after you do that, you can answer a few different questions like, for instance, which bacteria are present and at what relative abundance in that particular sample. And modulo all that stuff that we saw with the CAMI project, saying that a lot of the tools that do this are very varied in their performance characteristics. Let's say that we get this all completely correct. Now, what happens if you take a different sample from maybe a different environment or a different time point, and you go through this process again, and you figure out which bacteria are there, and at what relative abundance? How do you compare those guys together? So how do you compare these metagenomic samples? By the way, this is technically a research talk, but please, if questions, please ask, because we only make it through 2 or 3 slides, but we get very lively discussion. I would be very happy for that. Okay, so multiple samples. Let's say that you take like a nose nasal sample and one from the back of your hand, and you get all these whole bunch of short reads from it. And the sort of classical way that people go about comparing these samples is to, first off, classify them to figure out who's there, so generate a taxonomic profile, and then compare the classifications back and forth to each other. This seems like a very natural thing to do. However, what you find is that when comparing these taxonomic profiles, the current approach, classify and compare, oftentimes leads to very, very different answers. So there are a whole heck of a lot of metagenomic tools out there that will take in your short reads and give you these taxonomic profiles. And then you have a profile and nobody seems to agree on what metric you should use. There are biologically informed metrics, there are mathematical metrics, there are more information theoretic metrics, et cetera, et cetera. So if you take your selection of a method and a metric, and then do your further downstream analysis, you can get wildly different results. So it's kind of like a cartoon of this is your PCOA plots at the end of the day can just wildly go all over the place, depending on what you use. And so, yeah, please do.
So, those, and I know I should know this because your other talks and whatnot.
Uh-huh.
Those are all bacteria.
In this particular plot, these are all bacteria, but
one
yes,
level.
in a metagenomic sample, there are also viruses and plasmids and fungi and.
But that depends also on the level of resolution that you decide to adopt to do this analysis. So those could
Precisely.
be 15 or 1.5 million,
Yes.
depending on how fine you go in the.
And it depends on what technology you use. You can target certain genes that are only found in bacteria or only in fungus or prokaryotes.
Right.
Yep.
In fact, related question. If
Mhm.
one used this gene-based way of classifying,
Yeah.
assembling the data,
Sure.
essentially,
Mhm.
as opposed to the bacteria-based way,
Mhm.
are results equally unstable, equally variable, even if one takes the gene perspective as opposed to the species perspective? Remember there was this talk a few days back where these two were contrasted essentially. Does it make more sense to count abundances of genes or to count
right, to do a functional annotation instead? Yeah, so there are studies that show that the functional profile of metagenomic sample is less variable than the species identity. You still kind of get the same idea. You have some sort of these pie charts and a bunch of different methods to do it. So yes, you might have a little bit less variation for depending on which, what your data is, but yeah. Okay, so what is one of the reasons for this sort of unfortunate behavior? I like alliterations, so it comes down to distances, differences, and databases. So distances, there are a plethora of different metrics, and nobody really agrees on which one to use. The algorithmic approaches of each one of these different metagenomic approaches is completely different. Some can use marker k-mers, some use marker genes, some use machine learning, some use statistical models, etc. And so you can get completely different results, as well as the biggest thing is really relatively limited databases. So with each one of these, you're trying to answer who is there, so you have to compare it to something that you know. So you go back and you consult a database of previously gathered information. And this figure actually comes from our Metagenomics Journal Club that we've been holding here at CGSI, and I thought it was a fantastic figure. I had to share it. What this plot here, over here on this y-coordinate right here, are different samples or different locations of metagenomic samples. And over here is basically what percentage of the database that they were using explained the data that they had on hand. So for example, over here for human skin, more than 80% of that sample was explained by the database that they were using. And then you go over here to all these soil metagenomes, and you can see they're down in the low 1%. So yeah, so is in roughly the fraction of the sample that could be confidently identified as something that was in the database.
So 20% was not.
Right, yep. So this says 20% unknown, basically. Yeah. This is whole genome shotgun. Yeah.
And is it 20% of like different genomes, or is it 20% of like mass? 20% of.
Oh, that's a good question. Did they account for length? Is basically what you're asking. So is it percentage of cells and organisms, or is it percentage of just gDNA mass, basically? I don't know. Anybody from my journal club remember this figure, if they did account for that? Okay. Anyways,
obviously there's a big variability.
Precisely.
In the amount of coverage.
Exactly. And depending on what environment you're sampling from, you can have a very small fraction of your sample showing up in your database. Yeah.
Human skin, it can be like a lot of human genome, right?
Oh yeah, this is after filtering and quality control, stuff like that. Yeah. So they don't just literally take it and throw it on the sequencer. Yeah, so they do some filtration, stuff along those lines, and then after the fact, they align to the human genome to get rid of all those reads and take the unaligned reads and yeah, exactly. Yep. Good. So back in the previous slide where you're comparing classifications, if you took two soil metagenomes and you tried to classify those guys and then compare, you're really only comparing a very small fraction of the sample to another very small fraction of the sample. And that could be one reason why you see such large variability between different methods, etc. So what would be nice is if we could actually just compare these things directly to each other, instead of first off having to classify when we can only classify to a very small amount. Okay, so how are we going to do this? I'm going to do this with De Bruijn graphs. So, recalling what a De Bruijn graph is, we have a graph, nodes, edges. Our vertices right here are going to be k-mers of a fixed size. K is going to be different sizes and different points in this talk. And we draw an edge here if the tail of this guy matches over the head of that one. On this particular De Bruijn graph, you can see that our vertices have these disks of different radii right here. Those are going to be weights given by frequencies of these k-mers in a particular sample. So in this pictorial representation, there were a whole lot more ACTs than there were AACs in this sample. Okay, typically, how we've seen De Bruijn graphs used so far at CGSI has been to do assembly with this. So try to take one of these big dudes right here and find these paths in here so that you can then connect up all your k-mers and get longer contigs, and then do different things like maybe annotate them to find genes in them, etc. We've also seen Liorer-Pachter stuff with pseudo-alignment, where you take a bunch of these guys and you put them on top of each other, and then you have a short read and you basically align it to this De Bruijn graph. People actually also use these for genotyping and variant calling. So there's this program called Cortex by Zam Iqbal and Richard Leggett, which uses De Bruijn graphs as well. But we've already seen that, for instance, trying to take one of these guys and then do assembly with metagenomics is very difficult, especially for the low abundance organisms or the viruses and circular elements. This can be difficult. And you're losing information because this thing is a big old tangled mess and you're trying to detangle it into contigs. So instead of doing assembly, let's go ahead and try to compare De Bruijn graphs directly to each other. Because I'm basically thinking about this De Bruijn graph as a sort of more complete picture of what my metagenomic sample is than, say, an assembly, which can introduce errors and gives a sort of lower resolution view of my sample. There's Cortex that I mentioned previously. Okay, so it'd be great. It would be great if we took two soil metagenomes, and even though we don't know which organisms are in there, we could say, oh, by looking at the De Bruijn graph of this guy and of that one, these guys are more similar than those guys. So all I got to do now is come up with some sort of metric that will allow me to compare these De Bruijn graphs.
Of course, they should be generated with the same k-mers.
Yes. Yeah, same k-mer size. Yep. Okay, so how do we compare De Bruijn graphs? Well, there are a bunch of different metrics on graphs that you could use, but I was trying to come up with one that more faithfully represented these two graphs look similar to each other. So the first thing is that a De Bruijn graph of any metagenomic sample that you give me is this nice mathematical structure that is a subgraph of a full De Bruijn graph. So it's a subset of a full De Bruijn graph. What do I mean by that? Well, let's say that I have one sample and I take a bunch of k-mers of those guys. So like here, k equals 2, and you can't see those letters, but it's pictorial representation of the frequency in the sample. And that lives on some De Bruijn graph right here. Well, you notice that there are k-mers here that have no disk around it because these k-mers were not found in that sample. But mathematically, that thing still exists, right? It's still sitting out there. And so when you come along and you take another sample like this with different k-mer frequencies, you can slap it along on this guy, and these guys basically live in the same space, right? Great. Okay, so a very natural thing to do might be, well, how much are these guys overlapping? And if I had to shuffle these around, how much shuffling would it take to move these blue guys on the red guys, etc.? Yeah?
What is actually the motivation of using the De Bruijn graph structure? I mean, you could, in theory, just compare the frequency of all k-mers in this group and in this group, and you have some simple metrics. So what does the De Bruijn graph give you that you don't get just some counting vectors?
Yes, yeah, that is a great question. So, for instance, why don't you just, take these k-mer frequencies, those k-mer frequencies, and take the L2 distance between them or something like that. For a couple different reasons. First off is that the space that k-mers live in is not Euclidean, right? And so you have AAAAA and then AAAAC for a couple of k-mers like that. There's going to be a correlation between the frequencies of those guys in comparison to maybe a whole big run of T's. So there's this unique sort of dependency relationship between k-mers. What is that dependency relationship? De Bruijn graph. Yeah.
Because you overlap and you create that graph.
Yep.
It's got a lot more information than just the vector of frequency.
Precisely. Yep. The second motivation is that assembly, going from the De Bruijn graph or getting contigs, uses a De Bruijn graph. So it's a basic, not just mathematical object to study, but also biological, because it is the main data structure underlying assembly.
Is there an edge regardless of how much overlap there is? Like it could be one base or it could be two bases, or it could be 7 bases, and the edge is exactly the same?
Great question. So the edge for the Bruijn graph, all of it, so all of the tail, -1, so this is k equals 2, so it's very difficult to see. So let's go back way back to this guy right here.
Yeah.
These two overlap that guy.
Yeah.
So it's the entire k-mer -1
Yeah.
has to match. So for instance, like with this guy right here, the entire tail minus just the one character here has to match here. So it's k -1
Oh,
overlap.
So you do need that whole thing.
Yep, the whole thing to match. Yep. Okay, there we go. Great. Okay, so we have these things. We want to shuffle it around on this graph right here. We want the blues to overlap the reds. How are we going to do that? We are going to do that with this thing called the Earth mover's distance. Very aptly named. Okay, so what is the Earth mover's distance? Earth mover's distance is a very old and mathematical metric distance that has been rediscovered in many different fields over the years and used all over the place. So it's a distance between two probability distributions, first introduced by this French mathematician in the 1700s. Yeah,
Before you
please do.
get to that, so back to, let's say, issues of comparability, right?
Okay.
Whenever you're doing comparisons, there's this whole issue of how you normalize
Yes.
the data to
Yes.
compare. So here, these are frequencies. So,
Mhm.
for instance, how many reads you had from one sample or another doesn't really matter,
Right.
right?
Mhm.
Does read length matter? In creating these Bruijn graphs,
Mm-hmm.
does that matter at all? Probably not. It's just
Not-
something that,
Yeah, the only time read length would matter is when you're comparing extremely short read lengths to very, very long ones, because you basically miss one k-mer for
Yes.
every one once you terminate and move on. And so unless you're using read lengths that are roughly the size of your k,
yeah.
it's not really going to matter.
Okay.
Yeah.
Any other elements of, the way we sequence these samples that may actually hinder comparability?
So there's a lot of research into how, for instance, different kinds of errors affect the shapes of Bruijn graphs when you're using very large k-mer sizes. So for instance, if you have a sequencing error and you flip one of your nucleotides, that's going to create a bubble in these things, and there's various ways that you can go to try to collapse bubbles, or you have end splitting and things along these lines.
So if you were comparing two samples that had intrinsically very different levels of error
Yeah.
in them, there may be a consequence in the way that.
Absolutely.
Okay.
Absolutely. Yep. Yeah.
Maybe I missed this, but how do you take into account coverage? Like the total number of reads that you have in one
It's
sample, or what?
Yeah, so these are frequencies, so you just normalize by the total.
But of course, if you have higher coverage, they're more accurate.
Mhm.
That doesn't have an effect, or like has
It can, and it depends. So I'm considering this first off as an idealized, mathematical construct. So there's no statistical stuff stuck on top of it yet. So it's hard to answer a question of, how does this variability impact that when I haven't introduced that on top of it yet. But as unsatisfactorily as that answer is, basically, if you are comparing samples that are for very different, like sequencing technologies or different labs and stuff, you definitely do see a difference, and you can see in like, oh, it.
thinking about like different coverage you have like very high coverage in one sample,
Mhm.
in one mechanism of the sample, then you're going to see the very rare ones, very rare genomes. And so you're going to have very small frequencies for these, but you're not going to see the other ones.
Yeah, and for this, the impact of the low abundance ones, so the very low abundance frequency. So, for instance, if I had one of these guys that was really, really small, is the same penalty as that much mass on one of these guys. So it takes a whole lot of that really long tail to equate to a difference in one of these big hitters instead.
Let's say
So it's
that
not like L2 in that, that long tail can have a huge effect.
if you consider these as, instantiations of stochastic graphs of random variables, the two that you're comparing may have different intrinsic variability and
Yes.
the mechanisms that generate them. But the frequencies you take into account, right?
Mhm.
When you compute the distance that's weighted,
Yeah. Yes.
you move, but you have to wait for how big the dots are,
Right,
right?
right, exactly, yep. Yeah, and one thing one of my PhD students is working on right now is figuring out how this metric that I've yet to introduce is affected when these guys are given by random variables. So to figure out how that sort of uncertainty propagates through a Earth mover's distance. So Earth mover's distance, old distance between probability distributions. It basically tells you how much mass needs to be moved how far to be able to turn one of the distributions into another one. So from a super high level, we have a couple of distributions that look like this guy, and I want to take one of them and slide it over until it overlaps onto the other guy. Now, you can form this in terms of an optimization procedure. And the Earth mover's distance, I said that it's been rediscovered many times. It's actually called the Kontorovich, thank you, Rubinstein metric, the first Wasserstein distance, Earth mover's distance. So it has many, many different names. Yeah, but in any case, in terms of an optimization problem, we have two, and you can define this on more general spaces, but to keep things concrete, we have two probability vectors, so non-negative real vectors that sum to 1, and we have a matrix that's kind of like a joint distribution of these guys, because its row sums are equal to P, column sums are equal to Q, and it's also non-negative. And then we have some underlying distance metric. And then Earth mover's distance is going to be the minimum over all these matrices with row sums P and column sums Q. And then this guy right here, this is not a matrix multiplication guy right here. This is a Hadamard product. Basically, you take this matrix right here, you take your distance matrix, you stick them on top of each other by multiplying, and they just add everything up. And kind of think about this as an expectation, an expectation with respect to this joint distribution here, if you like thinking about it like that.
Could you please show for a second like the initial picture, the initial figures?
Right. Oh, it doesn't want to rewind my video. Sorry. PowerPoint. Ah, there we go. Hey,
Okay, cool.
I can't pause it.
But you actually don't move the intersection, right? The initial intersection you live with it.
Yeah. Right. So this is just one way to move it. This is not the most optimal way to move it. Yep, exactly. The Earth mover's distance is the most optimal way. Yeah, exactly. Okay.
And it's unique, the gamma that's solved.
Yeah, so the output that you get will definitely be unique.
Long moves.
Oh, yeah.
Many small moves versus one long move. It depends on the complexity of the code.
All right.
Yeah.
Thank you.
Yeah, the number that you get out is unique, but the flow, this little gamma right here, this matrix that achieves this minimum is not necessarily unique. Yep. Mass times distance. If gravity is equal to 1, this is work, right? So you can think about this as the minimal, so the most efficient way, or the least amount of work you have to do to transform one of these vectors into another one. Linear optimization problem. It's been pretty well studied. To be able to compute this thing, I'm going to be using this algorithm right here, which has complexity n squared log n and takes advantage of the fact that this distance metric has a very, very special kind of structure. It's going to be a graph distance. Okay. So EM de Bruijn is just basically doing this on a de Bruijn graph. So that joint distribution that I had has row sums equal to the Kamer counts, or Kamer frequencies, sorry, that should say frequencies, in one sample. Kamer frequencies in another sample right here. And this is what the joint distribution looks like, this big old matrix right here. And if you go and look and you see, like, hey, right here, this means that I moved 20 of these Kamers over to that, in the de Bruijn graph? Well, frequencies, but frequencies are a lot harder to read than integers, so this example is in integers. Our distance matrix right here, again, is the graph distance. So for example, graph distance 1, graph distance 2, the minimum number of hops you have to take to get from one vertex to another. And that's our distance matrix right here. And you can see it has a really special structure. You can take advantage of that when doing the computation. So reformulating our optimization problem for de Bruijn graphs, k-mer counts of frequencies in one sample, in the other sample, mass, distance, we're summing over the number of k-mers, minimal amount of work needed to transform one de Bruijn graph into another. Great, all fine and dandy. Does it work? Question?
Yeah, so the distance matrix in the case of the graph, the directed graph, is not really symmetric, right? Like moving from I to J, is not necessarily moving from J to I.
Right, yes, and I'm skipping over details because I wanted to get to the second part of the talk. I symmetrize this thing. If you see right here, it is actually symmetric, and it should be. Is it not? No, it's not. No, it's not. Okay. In practice, not in this example, but in practice, you just drop arrows, right? So you just make this thing undirected, and so you consider moving left or right on.
So in the Bruijn graph, it just allows to move from prefix to suffix or from suffix to prefix.
Yeah. Yep.
And then it becomes symmetric.
Yeah, and then it becomes metric. Yep. Yeah.
So can you give some sense of why this is a good metric? What does this quantity represent?
This quantity represents a minimal amount of work needed to transform one de Bruijn graph into another. It's a more natural metric to do when comparing k-mer frequencies or k-mer counts instead of any sort of Euclidean-based metric. Two de Bruijn graphs are the main object of study when you go and do assembly. And so this thing is basically a proxy for telling you how close or similar are your assemblies going to be if you were to be able to solve that problem.
But why this distance? So, okay, why the Bruijn graphs as summaries of the k-mers?
Right.
But why this distance and not another distance on graphs? No,
it's a it has many nice properties associated to it.
As a mathematician, what you should say.
Because
elegant.
Yes, exactly. Because it's beautiful. I like it. Yeah. Because I'm a mathematician, and these are easier objects to study. Yeah, exactly. Also, the proof is in the pudding, right? So, take a bunch of human microbiome project associated samples. This is 16S data right here. I'm going to do an all-v-all comparison for all these body sites, and I'm going to use a really small k-mer size because I don't want to sit around for a long time because it's n squared log n complexity. And using the algorithm that I mentioned previously, you can do this and get a PCOA plot that looks like this, and you see this wonderful, fantastic clustering into these like intratypes that were such the big thing when the Human Microbiome Project first came out. And actually, it clusters it better than these sort of industry standard UniFrac metric, biologically informed metrics. And we very much think, we know that these samples should cluster by body site because of the very distinct communities of organisms that live in there.
And the industry standards are reference-based?
Yes. Huh. Yes, exactly. Yep. Great. So this is all pretty cool, except as a mathematician, besides the metric being kind of cool, it was a little bit unsatisfying for me because I didn't get to do anything fun with this because because the computational efficiency, n squared log n, you can restrict yourself to only subsets of k-mers of any length and yada yada yada, but there's not a whole lot of hope to be able to reduce this computational complexity for this particular problem because of the loopiness of the de Bruijn graph. Just before I answer your guys' questions, we were able to come up with a linear heuristic algorithm to do this, but that's something for another talk. There's one, then two. Yeah?
So, I want to push you a little bit more on that question.
Okay.
I was not very happy with your answer.
Sure. Bring it on.
Sorry. for example, let me change the question. For example, L1 distance
Yes.
between two distributions
Yes.
have an interpretation in terms of supposed to draw samples
Mm-hmm.
from a distribution, and you want to test whether it comes from distribution P versus distribution Q.
Mm-hmm.
The L1 distance have a measure is connected to, for example, the probability of error of making a mistake.
Yep.
So I'm just wondering whether there is a similar type of interpretation from a statistical point of view of this particular distance that you define. So far you've defined a distance,
Right.
and it seems okay, but
Yep.
I don't really know what it means.
Right.
I'm not sure how I was going to use it. Because you want to persuade me to use it, right?
Yes, and I'm hoping the second part of the talk will do that too, because it turns out to be used by most every single metagenomics practitioner. UniFrac distance. So have you guys heard of UniFrac distance? It's like one of these top-sided papers, 5,000, everything like that. That's the Earth mover's distance. And
They don't know it, but
they don't know it, but they're using it. And so that's one thing. Second thing is, not being a statistician, I don't know any statistical interpretations of it. I do know of some probabilistic ones that we're working on currently, where you consider that instead of K-mer frequencies, they're given by some particular random variable with some distribution, and we're looking at seeing what the distribution of Earth mover distance values are going to be. And it turns out, since it's such a nice linear program, that's a tractable problem, and you get these really funky, cool distributions that are nice, pretty formulas. So you can either say that it's a tractable distance that has a nice interpretation in terms of work, or you could say that biologists are using it and they don't actually know it. So, yeah.
I have a question about actual frequency.
Okay.
So you show UniFrac on the left side.
Yes.
Is UniFrac basically the system that we do PCOA on that?
No.
So what is on the left side?
Oh, so UniFrac is a metric. I meant, yeah. So yes, UniFrac is a metric. You get all pairwise distances, and then you do a PCOA on that, and you get this. Yes, that is correct.
All right. So if I remember correctly, UniFrac basically assumes you have a phylogenetic tree, and then it does the arithmetic distance on the tree.
Yes.
Right. But then how do you know? I thought the whole thing is that you don't really know which tree goes to which
Correct.
part of the tree.
Correct.
Is it just
It's
part of the data on
unreferenced.
the left side?
Yeah, this is, so, that's one of the points behind this, is that these reference-based techniques, you need to figure out where those reads come on the tree, and that introduces lots of problems because of the databases and the metrics and the,
So follow up.
Sure.
Many people use UniFrac, people that I
Mhm.
work with, they use UniFrac, but without actually, but the distance they look, they build it through basically based on sequences.
Yes.
Let's say they do 16S, and they look at sequence of the sequence.
Yes.
They don't really care about the actual tree, they combine it with you could potentially do the same thing here. Sorry.
Yes. Yes. This is a fantastic segue. Exactly. So our second part of this right here is UniFrac. So UniFrac is exactly what you were saying right there. So it's a phylogenetic distance between samples based on a phylogenetic tree. So for a very simple example right here, I'm going to show you what UniFrac is in terms of unweighted. So let's say that these circles represent sequences from one sample, squares from another sample. We have some underlying phylogenetic tree that we trust, that we know, and then somehow we place these sequences on the trees. And then we want to somehow say, what's the difference between these guys? When this paper first came out, the way that it was calculated was as follows. For each one of these subtrees right here, you go and you count the circles, you count the squares, you take the absolute value of the difference. And then you go to the, so like in this case right here, we get 0. You go to the next subtree right there, you get 1 minus 2, absolute value is 1. And then you go and you do this for every single one of your subtrees, you get your answer, and that is going to be your UniFrac distance. Well, modulo normalization and some various other things, but this is how the algorithm was originally presented. 6 years later, mathematician and ecologist or statistician showed that UniFrac is Earth mover's distance. And this is a really beautiful paper that would be very fun to go through. But 29 pages, and I have like 10 minutes left, 5 minutes left. So unfortunately, you'll just have to believe that their proof is sound. And it's very cool. They didn't just prove this, they did some other things, like being able to introduce p-values and do some nice statistics with it. But however, they had no algorithms there at all, which was kind of didn't sit right. So while they showed the following, they didn't really give a way to calculate it. So this is the alternate view of UniFrac. So we first off saw with the tree over there with the circles and the squares. That's how it was originally presented. This paper right here is saying it's the same thing as the following. Here's a phylogenetic tree. We have branch lengths now on this guy. You have one sample, and you put pieces of that sample, either reads or fractions, etc., on nodes of this taxonomic tree right here. You do it with another sample, you stick that on there, and then you try to find the most efficient way to make the greens overlap the reds. So it's that same exact earth move resistance minimization problem right there. The most efficient way I'm demonstrating right here, and keeping track of how much it cost to be able to do this. So 1/3 went 0.1 plus 0.2, distance like that. And then that's the most efficient way to do this problem right here. And you get a number of 1/3. So the UniFrac distance here is 1/3. So here the interpretation being distance along the phylogenetic tree. Okay. I want to come up with an efficient algorithm to do this. I don't want to be stuck with n squared log n like we saw last time. I want to do this much quicker. Well, thankfully, this is a tree. A tree is a much nicer structure than just an arbitrary graph like a Bruijn graph. And so computing UniFrac turns out to be ridiculously easy. And so, and it turns out that this is not really new. This idea right here, it's been discovered and rediscovered in different areas, that to be able to compute UniFrac or earth mover's distance on this tree, all you have to do is take your mass and push it up. So here, they're already lying over each other, so we can make those guys disappear. Now these guys, you just take it and you move it up here. Oh, now they're overlapping. Get rid of it. Take these guys, push it up the tree. Now they're overlapping, disappear. Oh, hey, look at that. We get the same number. So you don't have to look for a most optimal flow. All you have to do is just push everything up, and you get your number. So boom, finished real quick. We got the UniFrac in linear time. What about this minimizing flow? Because, well, people have known now for a number of years that UniFrac is earth mover's distance. Earth mover's distance gives you more, right? It tells you this matrix of how to transform one of the distributions into the other.
But you said that's not unique, though.
It's not unique, but we'll find something that is unique with it.
Okay.
Yep. So it takes a little bit more for me and my PhD student to not only compute the UniFrac, but also compute this minimizing flow right here. So we prove that this little algorithm right here will give you this and compute the minimizing flow for you in linear time. And since we're also bioinformaticians, we decided to stick it up on GitHub so you can play around with it if you'd like. And it gives you that minimizing flow. So before I talk about what that minimizing flow will do, first off, let's note that, one, it's fast. Okay, fast UniFrac is the fastest implementation of UniFrac so far. This right here, number of leaves on the tree, time in seconds. This is EMD UniFrac right here. Algorithmic improvements can save you a whole lot, which is pretty cool. The other thing, not to brag, but I'm about to brag, this right here is written in pure Python because I'm not a programmer. I just sort of like hack together the script. This is written in C right here, and so you still get that. So, yay, algorithms for the win. What about this minimizing flow right here? Well, you get more for free. The flow tells you which organisms contributed to differences between your communities, how you had to transform one into the other one. But as I said previously, these are not unique. Well, it turns out that there is a quantity, flux across edges, which is unique no matter what your minimizing flow is. So we prove that this flux across edges is unique. So in this particular example right here, for example, we move this guy right on top of here. And so no mass went over this edge. And no matter what your minimizing flow is, that will always be true. Here, the flux across this edge right here was 1/3. And you can see that it doesn't matter if you have to move this guy here, or if you need to move that guy here, you will always be moving 1/3 of the mass across that edge. So that flux is true. We call this flux a differential abundance vector because it's telling you which organisms are differentially expressed between your couple of two samples. So, oh, that clock is 5 minutes ahead.
Yeah, that's okay.
Oh,
It's okay.
I've been like, yeah, okay. Slowing down slightly. Application to real data. So let's go ahead and take a couple of, or 40 twin pair samples. So let me ask real quick. Did that flux thing make sense? Flux cross? Cool, okay. So here is a PCOA plot using UniFrac or EMD UniFrac. They both compute the exact same number, so it doesn't matter which one you use. This is what your PCOA plot is going to look like. The paper from which I got this data hypothesized and later showed that there is a difference between microbial communities in these two samples right here. You just can't see it with this PCOA plot.
And these were twins.
Yes, yep, exactly. One of the reasons why you can't see it in this case is because UniFrac is just one single number, right? And your communities could be different in a bunch of different ways and still give you that same number. So let's instead look at either the flow, which might not be unique, but it's basically a partitioning of one of the numbers between one of the healthy samples and one of the ulcerative colitis samples that will tell you which organisms moved where when you had to move it along the phylogenetic tree. These flux quantities, represented here with differential abundance vectors, do a better job of explaining that. So for example, between the healthy samples as compared to the ulcerative colitis ones, there was more Bacteroides in it. You can just read it straight off of one of these plots just by looking at the flux across the edges instead of trying to look at just a single number that UniFrac is giving you. So it's kind of cool that you can get these things for free with not just no algorithmic hit, but a more efficient algorithm just by viewing UniFrac as the Earth mover's distance and looking at this minimizing flow sitting underneath it. So hopefully that's a convincing reason why you should look at Earth mover's distance in this case. Another question, yeah?
Related to Bruijn's question, so if you have problems with UniFrac because you missign to read maybe exactly the wrong species, you think this kind of addresses it because you're looking at, here you're kind of looking at different branches in the phylogeny and saying how much flux there was under the UniFrac distance on that branch. So slight errors in where you place things are hopefully less than a mistake.
Yes, exactly. So for instance, if you consider this right here as a clade way down on your phylogenetic tree, and you got, the rest of the tree sitting up there, yeah, if you mistakenly, assigned this guy to maybe over here, or some small change like that, that is not going to affect any of the flux across this edge right here.
It's robust to certain noises.
Yes. Yeah, and that robustness, of course, gets more and more robust the farther up you go along the tree. Yeah, exactly. Yeah.
So just to understand, so the flux is basically like snapshots while you compute the total.
Kind of. So there can be multiple different ways to be able to make the red overlap the green, right? I can move this guy here, or I could move that guy there. What it does is it tells you, no matter which way you did it, how much mass did you move across that edge? Yeah. So it's almost, so it is a differential abundance measure. So for instance, if that was negative and that was positive, flux across this edge will be positive a third, negative a third, telling you that there's a third more of sample 2 than sample 1 underneath this edge.
So, but then regarding to your question, then if you have problems with UniFrac because you completely mislabeled something, what this would give you is that you can see where it happened. But it will give you the same number of things, the same extent of them.
Yes, precisely, yeah. So right, garbage in, garbage out to a certain rank, right?
But you
So
develop some robustness. Or
yes.
some
Right. At some level in this tree, you will not have made an error. So for instance, you still got it to the correct phylum, maybe. So then your phylum and above fluxes will all be still exactly correct. And then below that, yes, it'll just reflect the error that you made. Yeah. Yeah.
I guess what I was referring to before is that in this case, the tree is fixed.
Yes.
I'm asking you, you start with the sequences. What do you think about CCMS and doing? If you start
Mm.
with the sequences, you build the tree from the sequences,
Mm-hmm.
and then you do UniFrac with the earth moving distance
Yeah.
between the two trees.
Mm-hmm.
Instead of doing it between two
With different trees, can you do it?
That's kind of my question. What would be better? Or is there a reason to believe one would be better? Or is there a coding difference?
Yeah, yeah. So the doing it along the tree, is there one that's better or not? So you need to have the same underlying tree for two samples, right? So if you have two samples, they have to live both on the same tree, or
Okay.
Sure.
A union tree.
Sure, yes, you can do that. Your interpretation will then be the similarity based on whatever that tree is. Doing this might not be as informative because that couldn't really be interpreted as a taxonomic or a phylogenetic tree.
But how would it compare to the Bruijn? Right.
Right, yeah.
So do you make more mistakes trying to infer a tree on which to do this?
Precisely,
Or
yeah,
do
because
you-
you have more choices to make. So I think the main difference is difference in interpretation, right? Because with the EM De Bruijn stuff, that's basically how similar would your assemblies be if you could solve the assembly problem, right? And then this right here, at least originally as presented, UniFrac was for phylogenetic similarity. So it depends on which interpretation you want your distance metric to reflect. Yes.
That's kind of your how you
Performance measure.
That's what I understood. That's how you measure. Not only you, people basically show it and say, okay, this is like feels nicer.
Right.
Right.
Yeah. Yeah. So of
To believe that the Bruijn graph is going to give you more like nicer pictures than just doing the two trees. Doing same distance, every moving distance. But now
You could try it on the same data.
yeah, yeah. So the reasons why I think you get a better picture with the De Bruijn kind of stuff rather than the UniFrac stuff is due to the fact that creating those trees, you have many different choices of what algorithms you want to do and all these parameters to tweak and everything along those lines, which introduces lots more of noise inside there, which is why it looks roughly like the same picture. It's just not quite as clean. And so yes, I do think the De Bruijn one is better than having to first off either take as truth of phylogeny sitting in the background or construct it yourself and then compare things. Yeah, so that would be my guess for the differences.
But from this approach, you get this flux, and you don't get the flux. Wait, can you get a flux for the De Bruijn?
No, unfortunately, because you have these cycles
Right.
in there, and you can go one way and completely miss. Yeah, exactly. Yes.
So my question was not sort of Earth movement distance
Okay.
in general.
Okay.
The coupling with the Bruijn graph.
Yes.
So I guess what is missing maybe a theorem which says the following.
Mm-hmm.
If you have two De Bruijn graph
Mm-hmm.
representation, and you want to go back to some assembly, you want to say that if it's close in your distance,
Mm-hmm.
the assembly, the reconstruction, is also close, right? I think that's what you want to say.
Yes. Yeah.
The
And
Bruijn graph itself is not a reconstruction; it's a representation.
Right.
Or intermediate step towards it.
Exactly.
right. Yeah. Assembly is an ill-defined problem.
There's no
Yeah.
theorem here, but I think that's trying to capture what it's trying to say.
Yeah.
Can I?
Yeah.
But it's not very obvious why that's the case.
It's not completely obvious, but
Yeah.
I think the issue is that there's no like the every two assembly basically two
Yeah.
that was one information, right?
So let's say it's a very rich
Yeah.
summary of the data. You don't go all the way to creating an assembly, which has a whole lot of complications, but it's
Precisely.
an informative, rich summary, certainly much richer than just a vector of abundances.
Yep.
One thing that I was curious about, so
Mhm.
you can show that there is one thing that is unique across all possible flows that minimize this thing.
Yep.
Very cool, so that you can get. Could there be other things that are unique? I'm thinking of this as a statistician. I think of a contingency table, right? You're fixing the marginals, and you have many distributions that give you those marginals.
Yes.
Right? In addition to this flux, could there be something else that's unique and that you could compute for free, as you were saying? Is this flux? What kind of animal is this flux? So to understand if it could have cousins that are unique as well in that
Yeah.
representation.
Number of non-zero entries in any minimizing flow. That's unique. This flux right here, those are the only quantities that we're able to tease out that are unique so far. Yeah. Yeah.
Did you want to say something else?
I guess that was it. There we go. You can use it for things that's fast. It might be cool, depending on your interpretation. Feel free to steal my code and improve it as you'd like. Cool. Thank you.
Is there anyone who has not asked questions?
Yeah, fantastic.
So in the tree, I guess the heuristic is like, pretty intuitive. You're moving up the tree to find, but in a Bruijn graph, like, how do you employ the heuristic because it's a very messed up thing,
Yeah. So first off, on the tree, that's not a heuristic, right? That's an exact way, and that is the way you do it. Yeah, on the De Bruijn graph, your heuristic is basically take the largest frequency and look one neighbor away, find the largest sink, basically. Put the mass there as much as possible. Do that for all the, from top down, and then move out step 2, and then move out step 3, like that. And then you're going to stop by the diameter of the graph, and these De Bruijn graphs are so interconnected, it's going to be.
Right, so both the algorithms in the end have the same computational burden. The heuristic version of the De Bruijn thing and the
Exactly, and it just turns out since the tree has no cycles, that heuristic is exact. Yeah.
And so, any idea of what the lower bound is n squared log n for the De Bruijn graph? Is that optimal?
So, I have a feeling, so as of yet in the literature, that's the best that's there. But the thing that is taking advantage there is bounded discrete distances. Or distance metrics that take on discrete and bounded values. So it's not actually taking advantage of the fact that it's on a graph structure. So the more loops or cycles that you add into one of these graphs, the further away it goes from n to n squared log n. And unfortunately, De Bruijn graphs are hugely interconnected. They're models for the fail-safe networks and stuff like that because they're so interconnected. So I feel like n squared log n is going to be the best that you can get, but I don't know. Yeah.
Thank you.
Cool. Thank you.